Now I'd like to take a moment to introduce Dr. Mary McMaster. She's been a regular speaker for us and very informative. She'll be speaking about the predisposition to WM, overview, and current updates. Dr. McMaster.
Thank you, Marlon, and thank you to Carl, the rest of the Board of Trustees, and the organizers of this meeting for having me. It's always a pleasure for me to be able to share what we're doing at the National Cancer Institute with Waldenström's patients. So it's important for me to say that everything you hear from me today represents my own views and is not necessarily representative of the official views of the National Cancer Institute. So as you know, Waldenström's is unique amongst cancers because it is a clinicopathological syndrome. Clinico meaning that it has clinical aspects that are unique to it, and pathological because it's partly but not entirely defined by what the pathologist sees under the microscope. You have to have not only the right kind of cells in your bone marrow or lymph nodes, but you also have to have this excess monoclonal IgM in the blood. One without the other is not Waldenström's. You have to have both. So turning now to the focus of my work over the past 20 years has been really focused on predisposition or susceptibility to Waldenström's, but through the lens of familial disease. And we had some really rather fundamental questions when we started out. Why does Waldenström's sometimes cluster in families? People came to us with maybe one family member who also had it, maybe 5. Why was this happening with such a rare disorder? At that time, also, there was literally no clinical research effort in Waldenström's around the world. And so we also viewed it at that time as an opportunity to learn more about the disease itself. Since 1998, which is when I first became interested in this, as you know, there are now many centers around the world that have a strong interest in Waldenström's, and so we have tended to move our focus back to the susceptibility piece. But we were interested if the disease process itself was different in families as compared to people who had it who did not have a family history. And then we wanted to know why were some people, irrespective of their family history, susceptible to Waldenström's in the first place? Was it a matter of genetics? Was it a matter of some environmental exposure that we didn't know about? Could it be related to lifestyle? So these were the things that we set out to try to answer. And to do that, we took sort of a multifaceted approach. So we recruited intensively high-risk families. We also have access to population registries, both within the United States and outside of it. So these have been extraordinarily valuable because where they exist, every single patient, when they are diagnosed, that diagnosis and certain core demographic information about that individual are entered into a database, and then the person is followed passively, but followed over long periods of time for outcomes. So in this way, it gets around the potential biases that are inherent in, say, a single center study. If I were to collect every patient that came to the NCI, I still couldn't say that my patient population was representative of everyone in this room, because not everyone in this room might have the ability to get to NCI, for many reasons. Maybe it's a factor of their disease and how severe it is. Maybe it's a factor of socioeconomic status. Maybe there are many, many factors that could affect why a person goes to a particular center or not. So these population registries are extraordinarily helpful because they give us a snapshot of all Waldenström's patients. We also use population genetics, so again, as distinct from familial genetics. And then we employ what's known as a case control approach, where we assemble a group of patients and then we find a group of people who do not have the disease, but who in every other respect reflect our patient population. So the same age range, gender distribution, occupation, et cetera. So we use all of these in different combinations to try to get at these questions. In our family study, what we have set out to do is to identify families with at least 2 individuals who have Waldenström's, or because in looking through the literature back in 1998, I found a handful of families that were described with 1 patient with Waldenström's, but a family member who had a related B-cell tumor. We thought, maybe there's a connection here. So we also were interested and have recruited families such as those. And then for comparison purposes, we have amassed actually a rather large collection of individuals who have no family history whatsoever. Of our participants, we ask that they fill out questionnaires related to their family history and their medical history, to give us permission to get their pertinent medical records so that we can verify their diagnosis and other parameters related to their Waldenström's. We ask them to donate a blood or saliva sample for our DNA and genetic studies. And then on occasion, in certain circumstances where there are particular questions that we feel like we need to lay eyes on that individual, we'll have them come to see us at the clinical center if they're interested. So what have we learned so far? That's really what we're here about today for. So the first question, how common is familial Waldenström's? So for this, we used our high-risk families and we went to population registries. And what we determined is that familial Waldenström's is rare, but it's not as rare as we first thought. We have about 100 families that we've identified and have actually enrolled in our study. I'm aware of many, many more who, for whatever reason, have not participated. But it's not nearly as uncommon as we had originally thought it might be. And we also know that in a referral hospital setting, about 5% of Waldenström's patients will report having a family member with Waldenström's, and up to 20% will report having a family member with a related B-cell tumor. So we thought that was very interesting, but again, remember what I just told you about a referral center and how there might be some biases associated with the patient population that is seen there. So we also went to population registries and in the Scandinavia, and in Scandinavia, not only do they register cancer diagnoses, but they also register relationships between individuals up to at least first and second degree. And so we were able there to show that there was a strong familiality in this disease. And then finally, we did a case control study with several thousand Waldenström's patients and over 20,000 controls. And again, we saw a strong relationship for familiality. But again, I say that, but I want you to realize that familial disease is still a very small part of Waldenström's overall. The average Waldenström's patient has no family history. So everything that I say today, you have to weigh it against that background. is that I'm telling you this from the perspective of this is my area of study, but please remember that the typical Waldenström's patient does not have a family history. So moving on, we wanted to know, do environmental or lifestyle factors play a role in Waldenström susceptibility? Here we actually looked in our families, we looked in the case control setting, and we looked in the population registries. And what we found, so Waldenström's, all cancer really, is considered a complex disease. A complex disease has many factors that contribute. And what causes the difficulty in studying and unraveling all of these and trying to determine, is this more important than that? Is there something that I can do that will change my risk? You have to understand, so this circle should have come up blank, so I'm going to have to trace it out with the pointer. But for instance, we know that genetic susceptibility plays a role in autoimmunity. So people with autoimmune diseases, as a rule, have a genetic predisposition. People with autoimmune disease, if it's severe enough, will get treated. That treatment itself and the autoimmune disease itself both can cause inflammation. We know that inflammation can cause immune dysregulation. And immune dysregulation, we believe, is sort of the underlying common pathway leading to the development of Waldenström's. But as you can see here, all of these factors are intertwined so that propensity for autoimmunity is related to gender, it's related to age. Immune dysregulation can be caused not only by autoimmune disease or inflammation, but age is related, genetic susceptibility is related. So you begin to see this complexity in this web that we're trying to tease out the individual bits, piece by piece. So how do we sort it all out? Well, when we're trying to determine whether one of these factors is actually causal as opposed to just sort of being along for the ride, or what we call associated, we have a number of things that we look at, and this list may vary a little bit depending on the particular factor that we're evaluating, because some things are more relevant than others. But in general, we want to know, did the presence of the factor precede the development of the disease? If you get Waldenström's and then you get rheumatoid arthritis, it's unlikely that the rheumatoid arthritis had anything to do with you developing the Waldenström's, right? We want to see that for a given factor, that people who are exposed to that factor, whatever it may be, are much more likely to have the disease than people who are not. And we call that the strength of the association. If you have more of the factor, if smoking is something we're interested in, if you smoke more, are you more likely to have disease? That would be something that would cause us to take that factor more seriously as a potential cause. We would like to see that it's reversible, that if you take it away, the risk goes away, or at least decreases. It should make biological sense. If it's a factor that affects your gallbladder, you have to stretch a little bit to make that relevant to your bone marrow. That's not to say it can't be, but those kinds of things at least enter into our thinking. We'd like to see several studies come up with the same results. That increases our confidence that we're on the right track. And finally, the specificity. So if you're exposed to this factor, how many outcomes might you have? If it's several, then it's harder to say, well, this is a cause of Waldenström's than if Waldenström's and maybe another B-cell tumor are the only outcomes associated with it. So for all the things we study, this is the thought process that's going on in the background. So do environmental and lifestyle factors play a role in Waldenström's? Well, both family studies and population studies of non-familial patients agree, yes, this is an important factor. And some of the specific agents that we have identified as being important include certain infections, autoimmune disease, family history, some chemical exposures, but we have only defined that very broadly. We don't have specific chemicals that have been identified yet. There's some hint that occupation may play a role and that lifestyle may play a role. So are there genes that predispose to Waldenström's? So for this, to answer this question, we have focused mainly on our high-risk families and population genetics. And for the bulk of my talk remaining, I'm going to be speaking about the genetic side of things because I find over the years that that's the piece that is the most confusing for people. And I'm going to try to lay it out. I hope I will be successful. It's complex. I will be here if you have questions throughout the meeting. Feel free to come. I'm happy to talk about it. But I find that people's understanding of how genetics works, and especially how it affects risk, tends to be uncertain. So I'm going to spend some fair amount of time on this. So if we go and take a biopsy of any place in the body and look at the cells under the microscope, we see that the tissue is made up of individual cells. And the cells have a particular organization. Within the nucleus of the cells, the genetic material is organized in structures called chromosomes within the nucleus of the cell. And if we then take one of these chromosomes and drill down and look at it in detail, we see that it's actually not this simple little X that you see in all the photographs. It's composed of a very tightly wound thread of genetic material. For those of you that remember old hand landlines with cords, it's like a telephone cord, and you know how they could get all wound up and tight. So the DNA is in this linear structure that's then spiraled around and then spiraled on itself until it's so tightly packed that it resembles a very smooth single structure. But if you unwind it and come farther down, you eventually see that it's made up of individual bases that are paired together to make this double helix form that we are so familiar with and hear about so often. So what can happen is that anywhere along this structure, and in human cells we have 23 pairs of chromosomes. So anywhere along this linear structure within any one of these chromosomes, something can happen that causes a change in one of the base pairs. And just to try to give a visual, here's your double helix, and here's your change. And then if we go back to the chromosome structure, you have a chromosome where somewhere along the chromosome, it could be anywhere, this change exists. But I think you can see from this that you could search for this change at the chromosome level, or at the DNA level, or at the individual base level. So for the purpose of discussing cancer genetics and susceptibility to cancer, it's important to understand that there are a couple of flavors of mutations that can occur. And in the scientific literature, we're moving away from the use of the word mutation because in actual fact, what a mutation is a variation. And not all variations are bad. Evolutionarily, the purpose of variation is to make us more adaptable to our environment. But occasionally, those changes don't, and they're deleterious. So a somatic variant is a variant that occurs in the body but not in the reproductive cells. So on the left, you have a schematic for somatic variant. So here's this woman, and she has developed a mutation in her breast. She does not carry that mutation anywhere else in her body. She may or may not develop breast cancer. But when she reproduces, her egg cells do not have that mutation. And so the chromosomes, the DNA in her offspring do not have that mutation, and the baby does not carry the mutation. This is distinct from what we call a germline variant. For a germline variant, the mutation arises in the reproductive cells. In females, the egg; in males, the sperm. They may have been born with it, or they may develop it during their lifetime. But once it's there, if by chance that egg or that sperm is involved in fertilization, then that variant is carried by the embryo in every cell of its body, and the child will have that variant in every cell of its body. So again, a somatic variant arises in non-reproductive cells. It cannot be passed on to offspring. None of the cells in the offspring will carry the variant. A germline variant is present in the reproductive cells. It can, but not necessarily. It's a 50-50 chance of being passed on to offspring. But if it is, then every cell in the offspring's body will have it. So just to touch on Waldenström's tumor genetics, I imagine that everybody in this room is familiar with the MYD88 mutation. And I just want to emphasize that this is a somatic mutation. It occurs in the Waldenström cells only. We are fortunate from a medical standpoint that it occurs in most cases of Waldenström's, because prior to its discovery, it could be very difficult to diagnose Waldenström's. But since it has been discovered, it has made it much easier to say with some conviction, you have Waldenström's and not something that's related to it. However, the key point on this slide is that it is not seen in the germline. It is not passed from parent to child. It is not a susceptibility gene. So again, what do I mean by genetic susceptibility? One or more genes that are important for normal cell growth will have changes, again, the synonym for that is variant, that affects the function of the gene. These changes are present at birth and can be passed from one generation to the next. But these changes may or may not be able to cause Waldenström's by themselves. In all likelihood, more than one genetic variant is needed in more than one gene, or you need a combination of both genetic changes and the right environmental exposures in order to get Waldenström's. So what does this mean? This means that there are probably a lot more people in the world who are carrying susceptibility genes who never develop the disease. So again, not to beat this to death, but I think this is such a critical, important point. Again, to review, only germline mutations can be passed down from parent to child. So on the left, we have our somatic mutation. The egg and the sperm are unaffected. The mutation occurs sometime after fertilization, either in the embryo stage or all the way up through adulthood. But only the cells that develop that mutation are going to be affected by it. And none of the reproductive cells will carry the mutation. In contrast, with a germline mutation, either the sperm or the egg will come to the union with the mutation. The embryo will carry that mutation or variant in all of its cells. And then later on in the germline cells, half of the reproductive cells will carry the variant. And I won't go into the reason for that, but it has to do with the fact that all of our genes we carry in duplicate. And at the time prior to the development of the reproductive cells, the duplicates split. So you'll have the variant in one, and the copy will not have the variant. But it's important to realize that in most cases, that variant is insufficient by itself to cause disease, and a second mutation must occur before cancer can develop. So the way we think about genetic susceptibility is that we have inherited variations that cause a change in one gene in one chromosome. This is acted upon by either other genes or environmental or lifestyle factors that might induce us to acquire a somatic variation in our bone marrow. And here is, so if this is a bone marrow cell, and you start with one gene change, then you get the second one, and that is what leads to the development of cancer. So to make this even more interesting, genes can have different effects. And we tend to, for simplicity, and this is really oversimplified, but it does help in sort of conceptualizing the idea. We call these genes with big effect. These are rare. And when I say gene, I'm actually talking about the variant, okay? Because all of our genes have big effects at one time or another during our development. They're all important. But a variant within a gene can have a big effect. These variants are very rare. If a person develops them, they lead to a high risk for disease. They may be modified by other genes, that's why you see these other DNA little spirals here, or environmental factors. But mainly the disease is developing on the basis of this single variant. So they have a very strong effect. And there's certain scientific strategies, experimental strategies that we use to try to detect those. And they're mainly looking at chromosomes, doing a type of It's DNA-based, but it's really a statistical analysis called linkage analysis, and then actually sequencing the genes and looking at the sequence of the gene itself. In contrast, we have genes with variants of small effect, and these are very common. Actually, up to 5% of healthy people walking around have one or more of these. We all have them. Everyone in this room has one somewhere, at least one, more likely hundreds. They lead to a small increase in risk individually. So in order to become ill, you have to have many of them interacting with each other. And environment plays a much bigger role in this scenario. And we have a very different way of investigating and looking for these kinds of genes that we call association studies, and I'll speak about that more in a moment. So again, another way of thinking about big effects and small effect genes. Rare big effect gene variants tend to lead to what we think of as simple genetic diseases, so one gene disease. They're seen only in the patients who have the disease. Rarely, if ever, are these variants seen in a healthy person. And again, a single variant can cause disease by itself. Complex disease, which is really what Waldenström's is due to common changes that can be carried by many people, where a single variant may have no effect whatsoever or such a small effect as to be insignificant. But when you put a lot of them together, then you have the potential to develop the disease. So how do we study genes with big effect? This is where our high-risk families are so incredibly valuable, because they're rare. And we see many people sometimes in a family that have the disease or something related to it. And so we know that that gene, whatever it is having a big effect. So what are we looking for? Well, genes contain the instructions to make proteins. And genes, I showed you that graphic some slides ago where they're made up of individual nucleotides all lined up in a row. And those together they make a code. And you can think of it as like instructions in a cookbook. Well, genes can have changes in spelling. Those nucleotides can be changed by variants. And depending on the type of variant, it will have a greater or lesser effect on the gene function. So here is an example. We have the word color. You can add in an extra letter, and if you're British, you haven't changed the meaning of that word one iota. On the other hand, you can change a letter out, and you've got a word that's very meaningful, but it doesn't mean what it initially meant. It's something very different. And then there are a whole host of changes that can be introduced that render that word completely meaningless. And so it has to be either replaced, or it causes the sentence to become garbled, or if you think in the cookbook analogy, if you put in 8 eggs instead of 1, you're going to have a mess. Again, we call these changes variants, and our genetic studies look for variants in genes. One of the key types of experiments we have used is called whole exome sequencing. This uses the patient's DNA, not the tumor DNA, because we're looking for these germline variants. We're looking for changes in the spelling of the DNA, It uses the fact that the same DNA spelling is going to be inherited from generation to generation. It's limited a little bit in that it only looks at the genes. We now know that the space between genes is incredibly important, but we're not quite yet scientifically at a stage where we can fully and accurately interpret what we see in between genes. So because we don't want to muddy the waters further, at present we're confining our investigations to the genes themselves. This approach is best for identifying rare genes with big effects. The studies are in progress, but I can tell you that contrary to what we expected when we started, we see lots of important gene changes in our high-risk families. But what we haven't seen so far is the same change across many families. Why is this important? Well, in contrast, in hereditary breast cancer, the BRCA1 and 2 genes account for not all hereditary breast cancer, but for a very large percentage of it. And so therefore, those 2 genes can be, if a woman comes with the right clinical picture that makes a clinician suspicious that she might have hereditary breast cancer, he or she can look at those 2 genes in combination now with some others and have a pretty high likelihood of being able to tell that woman yes or no, you have hereditary or you don't. In this case, each family is carrying its own private mutation. So the gene might be useful for that family, but it's not going to be useful for the next family. And the practical part of that is that it is exceedingly difficult to do this kind of work on a per family basis. It's also almost impossible to get companies interested in developing tests for a gene that only is going to be important for a small handful of people. We deal with that in the Waldenström's community all the time, right? Even Waldenström's generally has had to struggle to get the kind of scientific investment that it needs to progress. So those are the limitations. We're not giving up hope. We have only done a small number of families. We've recently included a lot larger set, and those data have just become available, and we're hoping to analyze them over the coming year or year and a half, and perhaps we'll find something that is more broadly applicable. But right now, it looks like for these genes of large effect, they're going to be private to a given family. What about genes with small effects? Well, here we can, because remember I told you that these small effect genes, all of us carry small effect genes. They're very common. So we can expand our sample size to include not only familial patients, but also non-familial patients. So again, we're looking for genes of small effect. And again, these individually lead to a very small or no increase in risk unless you have many of them acting together. So we use a strategy called an association study. And I like to think of an association study as mile markers. So you have a highway, here's our DNA. It's a long road. If we have mile markers, that tells us where along the DNA we are. And so just as if you ever listen to a police scanner or any emergency response, or if you've been in an accident or gotten stranded along the road, you say, I'm at mile marker X. And then people know where to find you, or a particular exit is at mile marker X. You know where to get off the highway. So they're linked. The mile markers are linked to important things in the DNA. Association studies actually look at the mile markers as a surrogate for the DNA. So we know the location of thousands of these mile markers in the DNA, literally thousands. Some studies include up to almost 1 million. Each one of these marks a gene or a gene region. And we can look at the genomes of many different people simultaneously because we're looking not at the individual DNA, we're looking at the mile markers, which is the same across everyone. And we look to see whether patients with Waldenström's have a particular mile marker more frequently than patients without Waldenström's. And that's what gives us our hint that something related to that mile marker is important in causing susceptibility to the disease. We have done previously family-based association studies, and we didn't study the entire genome in that exercise. What we looked at were specific genes that we knew were important in the immune system. If you remember earlier, I said we believe that immune dysregulation is sort of the underlying common pathway in Waldenström's. We found a few genes that contribute to risk, but those need confirmation. More recently, we have done what's known as a genome-wide association study. In this case, we look at the entire genome using these mile markers, and we compare the marker results for many different Waldenström's patients with those from healthy people. Our rationale was that Waldenström's clusters in families. We know that relatives of Waldenström's patients have increased risk for Waldenström's and B-cell disorders, and that we'd not been successful in identifying yet common rare gene variants. So we felt that a study that was enriched with familial cases might actually also be enriched for these common susceptibility genes. So whereas most of these studies require thousands of patients, we had 200. We did have 3,700 controls. We did the analysis, we accounted for age because we know this disease is age dependent, it's also sex dependent, and it also, their ancestry is important in terms of race, not heredity per se. We then replicated the study to see, so we got some findings, and we said, okay, we found these findings in a sample that was largely familial, but is this relevant for people who don't have familial disease? So we got together another set of Waldenström's patients that were did not have a family history of Waldenström's and did a similar analysis. The results are that we found at least 2 regions of the genome that can contribute to Waldenström's susceptibility. There are probably more yet to be discovered if we can put together larger samples of patients. We don't yet understand how this happens. We're trying today. I left NCI yesterday after a discussion. We're trying to figure out why these particular regions are important. But it's more evidence that Waldenström's likely results from a combination of events, not a single gene that is passed from parent to child. So I'm running out of time, but are family members of Waldenström's patients at risk for other conditions? So here We have gone to our registry studies with thousands of patients, thousands of controls, and we see that several other B-cell disorders are much more common in these families than they should be. When we see MGUS, it is almost always IgM MGUS. And there's some hint that non-monoclonal IgM, but just an elevation of IgM, may be more common in families as well. Quickly, I'd like to go through this concept of risk. Are you more likely to be killed by a shark or coconut? And most people say a shark. We all carry, this idea of sharks as teeth, but in fact, a coconut is more likely to fall out of the tree and conk you on the head and kill you that way. And then there's the concept of relative risk and absolute risk. So we hear about the things in the news every single day, and it's different from day to day. But just for an example, let's just say that if you don't drink caffeine, you have a 1% risk of having a heart attack. But you have, say, a 1 in 3, 30% chance of having trouble falling asleep at night. If the relative risk is twofold or double, and when you drink 3 cups of coffee, then what that means is that your chance of a heart attack goes up to 2%. That's only a 1% change in your absolute risk. Whereas insomnia, which was more common to begin with your 3 cups of coffee, you double that to 60%, you've had a 30% increase in your absolute risk. And the point here is that when you read and when you hear those of us get up here and talk about, oh, I have a family history, my risk is so much higher. But Waldenström's is such a rare condition that yes, it may be, with no family history, the risk of Waldenström's is 0.5/10,000%. Whereas with breast cancer and prostate cancer, it's 12 and 11% respectively. With a positive family history, let's take worst case scenario. I'm 20 times more likely to develop Waldenström's if my mother had Waldenström's. Well, my risk just went up to 1/100th%. That's still pretty low. Now, if it were breast cancer, we're talking some real changes in risk here, okay? Because my risk at just twofold goes up to almost 25%, or 1 in 4. So in thinking about family history and the risk that to our children, to our brothers and sisters, we have to keep that in mind. The actual absolute risk is not that great. So again, how do we think about Waldenström's risk in 2018? We're coming to the understanding that lifestyle, genetics, environment all play a role, and that where these interact, the risk increases. So our take home for today, a single gene is not likely to account for Waldenström's in all families. We found linkage to more than one region. We've found associations to these small effect genes that I mentioned, and there's evidence suggesting that environment may play a role. The risk for Waldenström's depends on a combination of genes, environmental exposures, and possibly lifestyle factors. Should healthy people get screened? So this is a question I get asked all the time. The answer is maybe. They are at low risk to develop Waldenström's. So to get screened because they might develop Waldenström's later in life, I'm not so sure that that's a good idea. But there are new data coming out that we didn't discuss today that suggests that MGUS alone may have health effects apart from cancer risk. So this is an area of active research, not by me, but by many people in the field. And I think that might color our recommendations in the future, because if you are screened, I mean, what we screen for is MGUS generally, because that's the risk factor, the precursor condition for Waldenström's. But in the past, we said, well, you have MGUS, there's nothing we can do for you, we're just gonna watch you, it could take years, you may never develop cancer, so why bother getting screened in the first place? But if MGUS actually carries some health effects of its own, then there might be more of an argument to go ahead and screen people. If, for whatever reason, a person is screened and does have MGUS, they need to be followed indefinitely. They may never develop Waldenström's. They may never develop multiple myeloma or CLL, but there is a risk. And so if you're going to go down that path, you have to be prepared to be followed then indefinitely. And finally, these results need more study and the use of new approaches, and we're blessed that every day new technologies, new bioinformatics approaches are coming online, and so our tools continue to expand, and we're very hopeful that these remaining questions will continue to be answered over time. But we still need your help. It's not, the story hasn't come to the end yet. Help us get there. There are so many ways that you can contribute. So I'd like to thank you. I know I've run over a couple minutes, but it's been my pleasure to be with you, and I'm happy to take questions.
Is this on?
Yes.
So I was wondering, what would be the health effects of MGUS that you're speaking of?
Well, there are a variety of them, and as I say, this is an emerging area, but MGUS has been shown potentially to be associated with things like bone fragility and risk of bone fracture. There's some data that suggests that it might be related to cardiovascular events. So this is all very new. It's growing. I think that, and it's not my area of research, so I'm not right at the top of my game in terms of being able to answer those questions. But the key point is that until recently, MGUS was thought to be just something that was in the background that, other than for its risk for cancer development, had no other ramifications. But we're learning that that may not be true. Yes, ma'am.
Thank you very much. You're a really good teacher, a really good teacher.
Thank you.
I have familial Waldenström's. I have a first cousin with Waldenström's, and my brother had CLL. We would love to participate in something, give you data, whatever. Is there a way for us to do that?
Well, thank you. Please feel free to seek me out. I can give you information about what we're doing at NCI. Thank you. Yes, sir.
Again, great talk. Thank you very much. I was wondering more on the tumor side rather than the host side, but I haven't heard anything in this meeting yet about the trisomy of chromosome 4, and I'm wondering, it's common in Waldenström patients in the neoplastic cells, and I'm wondering if there might be some insight that geneticists could get from that trisomy.
I think you may be onto something there. It's always been interesting to me that in the very first genetic studies that we did involved linkage, and we found linkage to chromosome 4, in people who did not have trisomy 4, suggesting that there might be a gene on chromosome 4 that, when it is duplicated, in the trisomy. So trisomy, for those of you that don't know, trisomy is 3. We ordinarily have 2 of each of our chromosomes in each cell, but if a piece of the DNA is duplicated for whatever reason, then you can get more than one, or a piece can be deleted, and then we call it monosomy for that chromosome region. So when you see duplications and deletions, that's a clue that there's something in that area that's important. What's not as clear is whether it's related to the disease progressing, so it's relevant to the Waldenström cell alone, or whether it has relevance for the entire person. We don't see, when we see trisomy 4 in Waldenström cells, we don't see trisomy 4 in skin cells from that patient, or heart cells, or lung cells. So it's clearly not a susceptibility issue, but it must have something important to do with the progression of the disease.
The thing that catches my eye is the number of chemokine and chemokine receptor genes in chromosome 4, and also c-KIT.
Yes.
Just wondering if they may have something to do
Yeah,
with it.
I think that very, very likely may be important. And it may be that these chemicals are either driving the tumor growth, and so having more, the tumor benefits from having more of them. So, but it's a very interesting finding, and actually we've known about trisomy 4 for a long time, but I think we didn't fully, a lot of the duplication occurs at the subchromosomal level, and we didn't appreciate how often it was there.
Thank you.
My family has a lot of different kinds of cancer. My mother and all her siblings died of different kinds of cancer. And I have had breast cancer and a recurrence 30 years ago. I had a very rare cancer in my uterus about 20 years ago. 2 years ago, I was diagnosed with melanoma stage 3, and 1 year ago, they told me that since 2006, I've had an elevated IgM. So now I'm in treatment for Waldenström's, and I just wonder if you see a lot of families like this with so much different kind of cancer. I've had genetic studies done at the University of Washington, as my sister has, and they say I have no connection. I don't even have BRCA1 or 2.
Okay. Did you have individual gene testing, or did they do a panel?
A panel with all our relatives, yeah.
Okay. So we occasionally, some of our families have that kind of a history. Sort of a side interest of mine is multiple cancers in an individual who has Waldenström's, although my colleagues who are interested in that field, they're interested because they're worried about the effects of treatment on later risk of cancer. So, for example, we know that people who've had radiation for Hodgkin's disease in the upper body, if they're female, are at risk to develop breast cancer later in life, that sort of thing. From my perspective, it's more from trying to understand the susceptibility angle. So could there be genes that are relevant for a variety of cancers, including solid tumors? Now, it's interesting that you say melanoma, because melanoma falls within the umbrella of what we consider immunological cancers, just as Waldenström's is. And so anecdotally, we do see an increased connection between melanoma and Waldenström's in the same person. Within my families, I see a lot of melanoma, but I've never done the formal analysis to show that it is truly increased and not just, my sense that it's increased.
Okay.
But there are other cancer syndromes that include non-Hodgkin's lymphoma, and of course Waldenström's is a form of non-Hodgkin's lymphoma, but none of them include Waldenström's specifically, at least so far as I'm aware.
Okay. So would it be helpful for my children to have any more studies done, or to have my children done?
No. In this kind of situation, unless and until the causative gene is found in your family, and the person it's going to be found in is a person who has cancer, then it's really not helpful at all to screen the children, because a lot of these changes, even in well-established genes like BRCA, a person can have a change in a BRCA gene and they can have breast cancer, but it does not automatically follow that gene change caused the breast cancer. There are plenty of what we call variants of uncertain significance and even benign variants within the BRCA genes, and it may be just chance that those occurred in someone who has breast cancer for another reason. So to then go to someone who doesn't even have the disease and look for changes, you can imagine the complexity that introduces.
Okay. Thank you very much.
Yes, indeed.
Given the foundation that you created today on the interaction of environmental factors and other factors, is there any work being done right now on the effects of environmental radiation exposure and the development of Waldenströms? And if not, do you see that potentially on the horizon in the future?
Not to my knowledge. So far, the biggest impediment to studying environmental risk factors is that in order to reach definitive conclusions, so statistically valid conclusions, you typically need hundreds, if not thousands, of patients and thousands upon thousands of controls. And in the Waldenström's research community so far, we have not been successful in amassing those kinds of numbers. I do participate in a consortium that is the overarching goal of that. It's international. It's composed of researchers from around the world whose common theme is that they're interested in lymphoma of all types. And so we have all pulled our Waldenström's patients together and collaborated together to try to get the kinds of numbers able to do these kinds of studies. And we've done one, and I referred to that was actually the one that had all the pictures on it with the viruses and whatnot. Radiation did not fall out as important, but again, that study might have been underpowered to detect an effect. So I think that in the research community, we keep pushing for collaborations to try to pool data, to try to pool our common knowledge and our common resources, because that's the only way we're going to be able to answer that kind of a question.
I don't know about Waldenström's, but I've read that some types of cancer are increasing. And when you were describing genetic changes that happen, it occurred to me, is there any chance that as the human race goes on, generation after generation, that genetic changes accumulate and build up in subsequent generations?
So the answer there is yes and no. Genetic variation from the purpose of it, if you will, is to improve the organism's adaptability to environmental pressure or genetic pressure. And so the variants that are retained because either A, they don't affect the organism early enough in life that it dies before it can reproduce. So really bad mutations, those are the ones that lead to miscarriages and early childhood deaths and that sort of thing. And those individuals that carry those don't live to reproduce, so they can't pass them down. But these changes they can occur spontaneously. So we didn't all get them from our ancestors. They're occurring in us every day because of radiation exposure, and we think about medical radiation exposure or atomic bomb radiation exposure, but I flew in an airplane here yesterday, and so I was up above the layer of the atmosphere that protects me from radiation in the sun. I got radiation exposure there, and so cumulatively over time, I may develop a mutation related to that. So mutations come and go, but in general, the ones that stay that have an effect, it's because either they're having a net zero effect on that individual's adaptability, or the species adaptability. Really, we're talking about the species level here. Or it helps something to survive.
So it
Cancer
can be good then.
Which can be good, exactly. That's exactly my point. So early on was that that's why we're moving away from this notion of mutation, because genetic variation is really net balance, a good thing. But cancer has co-opted genetic variation. It's good for the cancer. It's not thinking about the rest of the organism, its host, right? So for the organism, it's not a good thing.
Thank you.
That's all the time we have for questions. Let's give a good round of applause for Dr. McMaster.